Hello everyone. Today we are going to discuss the topic from 4.3 Nucleotide Sequence Databases. In earlier classes, we have discussed regarding the nucleotide sequence databases in brief. Now we will discuss these databases in detail. This NCBI, that is the GenBank, then EMBL, and the what DNA data bank of the Japan. These are the three nucleotide sequence databases, and these biological databases are divided into these four: sequence, structure, genome, and specialized databases. Among them, we have to discuss today the nucleic acid or nucleotide sequence databases. And these types of databases, among these, the sequence databases, the what we have to discuss the nucleic acid databases or the nucleotide databases. Among them, the gene bank, then DNA data bank of Japan, and the what EMBL, European Molecular Biology Laboratory data bank. All that you have to discuss in detail. The sequence databases, and in the sequence databases, we have to discuss that there are the two types. One is a nucleotide database, and another one is a protein databases. In this class, we have to discuss the what nucleotide databases. The biological information of nucleic acids is available as a sequences, while the data of proteins are available as a sequences and structures. Sequences are represented in a single dimension, whereas the structure contains a three-dimensional data of the sequences. The database is complemented with a generalized software for processing, archiving, querying, and distributing the data. And such databases consisting of nucleotide sequences are called as nucleic acid sequence databases. Means those databases having the what nucleotide sequences. These are called as a nucleic acid sequences or called as a what nucleotide databases. Among them, these three important nucleotide databases, that is a gene bank, that is governed by or that is by the what that is NCBI, then EMBL and the DDBJ. These are the three important what nucleotide databases that we have discussed. That we have to discuss. That is the what the gene bank from NCBI, EMBL, that is the what European Molecular Biology Laboratory, and that is governed by the what that is a this maintained by the what European Bioinformatics Institute, and the what that is a DNA data bank of the Japan. These altogether, GenBank, EMBL, and DDBJ is under the database International Nucleotide Sequence Database Collaboration. These all are three are the primary databases. This is the what the composite database, that is International Nucleotide Sequence Database Collaboration. There are three cheap databases that store and make available raw nucleic acid sequences to the public and researchers, and these are GenBank, then EMBL, and the DDBJ. This EMBL stands for the European Molecular Biology Laboratory Database, and the DNA Databank of Japan database. And these three are the what primary databases, and they are also called as an archival database since they are the repository of all nucleic acid sequences. They are populated with experimentally derived data such as the what nucleotide sequence. GenBank is physically located in the USA and is accessible through the what NCBI portal over the internet. The EMBL, that is what European Molecular Biology Laboratory, is in UK, and DDBJ, that is the DNA Data Bank of Japan, is in Japan. All three accepts the nucleotide sequences submission and then exchange new and updated data on daily basis. Means they exchange all these three, GenBank, EMBL, and DDBJ. They exchange the data daily, and the new and updated data on daily basis to achieve the optimal synchronization between them. There are three databases these three databases are primary databases as they house the original sequence data. They collaborate with what the sequence read archive, that is SRA, which archives raw reads from the high throughput sequencing instruments. Then this is the web homepage of the what NCBI, that stands for National Center for Biological Biotechnology Information, and from where you can access the what that is a various databases. That is mainly the gene bank. It is what that operated by this. Here you can observe the what the PubMed also, that is a literature database. This blast is a word, the term search engine. Then the word is omim, this is the word the another that is a word the database related with the online Mendelian inheritance in man. All these are the what the different types of the word. This is the word the gene bank related with the word that is the sequence literature databases. This is the word the where the what you can you can observe the word that is a home page of NCBI. Then the standard contents of the sequence databases. If you observe the what that is a standard sequence of the databases, it having the sequences. These are the sequences. Then it having the what the accession number. Each and every having the what the accession number. Then it having the what references. Then taxonomic data means which source. There this is a written that is what the Saccharomyces cerevisiae. This is the what the taxonomic data. Then annotation and the what that is a curation. Then keywords, then cross references and documentation. All these it having. So, and this is the what the entries. This is the fly base is the what that is another database related with the what that is a fly or the that is a Drosophila. So all these are the word that is like that. These are the word the web pages or how you can site. This is the NCBI NLM site, means this is from the what the we are assessing the what the gene bank, that is from the what that is a NCBI. Then this primary database is or the nucleotide sequence databases, that is the gene bank, EMBL, and the DNA data bank of Japan. It is a GenBank is a US primary nucleotide sequence database established in 1988 and maintained by the NCPI, that is the National Center for Biotechnology Information, and it is a very comprehensive biological database and provides 42 different resources. It provides a simple and easy to use web interface. And Trace is a retrieval tool or search engine for this GenBank. This is the what the web page of the what that is a website of the what that is a this gene bank at the NCBI. Then another one is a EMBL, that is European Molecular Biology Laboratory, and it is a Europe's primary nucleotide sequence resource established by the EMBL and maintained by the what that is the EBI, that is what European Bioinformatics Institute. And SRS is the what that is the sequence retrieval tool, is a retrieval tool or the retrieval engine for this EMBL. And the DNA Data Bank of Japan, that is a DDBJ, is mainly collect the data from Japanese activity but accept the submission from all researchers in the country. And this is the what the web page or the home page for this or the website for the what this DDBJ. First is the what the gene bank. The GenBank is a US primary nucleotide sequence database established in 1988, which is maintained by the what National Center for Biotechnology Information as a part of the International Nucleotide Sequence Database collaboration. Means this international nucleotide sequence database collaboration is a what that is it is a composite database and it comprises of all three, that is a gene bank, EMBL, and the what that is a DDBJ. Then gene bank is a collection of all known DNA sequences from scientists around the world. It is very comprehensive biological database and provides 42 different resources. The GenBank sequence database is an open access, annotated collection of all publicly available nucleotide sequences and their protein translations. GenBank receives sequences produced in laboratories throughout the world from more than 1 lakh distinct organisms. It provides a simple and easy to use web interface. This is the website of this, the NCBI, from where you can access the GenBank. And for this, the research engine is a what? Entrace is a search engine or it is a retrieval tool. The GenBank has become an important database for research in biological fields and has grown in recent years at an exponential rate by doubling roughly every 18 months. Means it having the huge data or the sequence database and it is increasing day by day by the what that is by the exponential rate, and the rate of doubling the data it is that is in every 18 months the data gets doubled. Then searching the gene bank is fairly straightforward and can be done with a variety of search tools. Searching the gene bank include the BLAST, that is basic local alignment search tool. This BLAST it searches the DBEST, that is what database of express sequence tag. Then DBSTS, that is a database of sequence tag sites, and you can also submit the sequences via the www with a bank kit. This is the software by which you can submit. So these all are the what that is a searching the gene bank, that include the BLAST, DBST, and the what DBSTS. Then another word that is a nucleotide sequence database is a EMBL. That is a European Molecular Biology Laboratory. It is a Europe's primary nucleotide sequence resource that was established by EMBL and maintained by the European Bioinformatics Institute. This EMBL or European Molecular Biology Laboratory database is a member of the International Nucleotide Sequence Database Collaboration, which includes all these three, that is a primary databases, that is a DDBJ, EMBL and GenBank. EMBL receives the database from large-scale genome sequencing projects and direct submission from individual scientists and sequence data extracted from biotechnology patent applications to the European Patent Office. Then the SRS is a sequence retrieval tool for this EMBL. Means this EMBL it receives the what databases from where the sequence large genome sequence project and the what the scientists and the what that is a patent office from the what that is a European countries. Then next is the what the submission to the EMBL nucleotide sequence database. For the submission purpose, Webin is a preferred system. The Webin is the preferred submission system for the nucleotide sequence and biological annotations, including third-party annotation and the alignment of the data. Webin has been designed to allow the rapid submission of single, multiple, or very large number of sequences or bulk sequences and is available. And its website says that is address is http www.ebi.ac.uk, and that is what EMBL submission webin.html. This is the website for this. Then the third one or third primary nucleotide sequence database is the what that is a DNA data bank of Japan, or that is called as a what DDBJ. The DDBJ, the DNA data bank of Japan, was established in 1986 to be one of the major international DNA databases with GenBank and the EMBL. It is located at the National Institute of Genetics, that is NIG, in the what Shizuoka prefecture of the Japan. It is the only nucleotide sequence data bank in Asia. Then mainly it collects the word DDBJ collects the data from Japanese researcher, but it accepts the submission from any researchers in any country or from any country. This is the what the website address for this DDBJ. That is the what HTTPS dot then the say then slash double slash www.ddbj.nig.ac.jp. Then there are the secondary databases of the nucleotide sequences. Many of the secondary databases are simply sub-collection of sequences culled from one or other of the primary databases such as GenBank or EMBL. There is usually a great deal of value addition in terms of annotation, software, presentation of the information, and cross references. There are other secondary databases that do not present sequences at all, but only information gathered from the what sequence databases. Among these databases, that is what Omniome database is what the secondary database or secondary nucleotide sequence database. This Omniome database is a comprehensive microbial resource and it is maintained by the TIGR, that is TIGR, the Institute for the Genomic Research. It has not only the sequence and annotation of each of the completed genome, but also has associated information about the organisms, then structure and composition of their DNA molecule, and many other attributes of protein sequences predicted from the DNA sequences. It facilitates the meaningful multi-genome searches for analysis. For instance, alignment of entire genome and comparison of the physical proper of proteins and genes from different genomes, etc. The next database or secondary database for the nucleotide that is a fly-based database. This fly, that is what Drosophila fly, Drosophila melanoblaster. It is a consortium sequenced the entire genome of the fruit fly, that is Drosophila melanogaster, to a high degree of completeness and quality. Then next, that is a what database, that is a what ACEDB. It is a what is this ACB? That is what the Sinorhabditis elegans. What it is a repository of not only the sequence but also the genetic map as well as phylogenetic information about what Sinorhabditis elegans. It is a what nematode larva generally utilized for the study of the what the genetics or used in the study of the genetics and the developmental biology. And this database it is of the what the Sinorhabditis elegans, that is a nematode worm. That ACEDB, that is what the sequence that is a secondary database or the sequence that is a secondary nucleotide sequence database. Then sequence submission is done by using the Bankit or Sequin. These are the what the submission algorithms. Then the search engine for data retrieval for these is the entries. And it retrieves the information across all the resources under the NCBI. Example, the what the PubMed, then taxonomy, SNP, then PubChem, etc. Then what are the tools of the what that is the analysis of these nucleotide sequences? That is the one is a BLAST, then primer BLAST, B-Link, ORF Finder, and the Genome Workbench. Among them, we will observe the what is this BLAST. This is the tool for the analysis. The BLAST, what is the role of this BLAST? BLAST, it was developed by the Stephen and its colleagues at NCBI in 1990, and this BLAST compares the sequences. This what is this NCBI? That is the National Center for Biotechnology Information, and the long form of this BLAST is what the Basic Local Alignment Search Tool. And this BLAST takes the query sequence And to this query sequence, it compares it with millions of sequences in the GenBank databases by constructing the local alignments. And after that, it lists all those that appear to be similar to the query sequence, and that we called it as a hit list. And it tells you why it thinks that they are homologous or similar. And blast give us the suggestions, and you have to come to the conclusion by that suggestion. And website for searching the GenBank and other NCBI sequence database is this, that is the www.nlm.nih.gov/blast. And it can be used to search the NCBI nucleotide database, that is the DNA sequences, as well as the NCBI protein databases. So this is the BLAST tool, which is very much useful for the what that is a sequence analysis, or the what that is a nucleotide sequence analysis. This is all about the idea regarding the what that is a nucleotide sequence databases, or about the nucleotide sequence databases, that is the primary as well as the secondary and the composite databases. Thank you.